I'm working on a new type of programmable material, and it is analogous to DNA nanotechnology. And so we're basically building off of everything that we've learned in DNA nanotechnology over the last 40 years. But our building material is a synthetic mimic of DNA, and so it's called peptide nucleic acid. In the realm of modern manufacturing, biomanufacturing, and sort of micro-robotics, where I'm working, we're really excited about DNA nanotechnology. And so we have numerous avenues in our lab working on novel nanobiosensor applications. We're also really interested in using those DNA structures as kind of like adapters or chaperones to facilitate larger scale construction. So we also have sort of a micro-robotic thrust where we've got this top-down assembly married with bottom-up assembly. Overall, what we're really excited about with nanostructures made using peptide nucleic acid is that they have potentially really exciting benefits that differentiate them from existing DNA nanotechnology, but they also have all of these unusual and new leverageable traits. DNA, when it enters the body, when it enters your cellular environment, is attacked by nucleases. And so they take it apart, and that's a natural defense of the body to prevent viral DNA from getting into the nucleus of a cell. But that means that your body breaks down DNA nanostructures really effectively. And so one thing that's really interesting about peptide nucleic acid is it's basically a very elegant Trojan horse. The body doesn't recognize it. So if we can use it in certain instances to replace DNA nanotechnology, there's never going to be a fear that it's going to be broken down. So if we send in a sensor or a diagnostic or a therapeutic, it won't be taken apart. And that's really critical. And then one other thing that is really exciting for us is that you can actually pre-organize these structures to be left-handed. And so this is work out of Daneth Lee's lab at CMU. And there's a really special feature of left-handed PNA, which is that it can't bind to DNA at all. So not only would they be the Trojan horse type molecules, but there'd be no possibility of them cross-hybridizing with the DNA in somebody's body. And what they found is that right-handed PNA actually binds to DNA more strongly than even PNA. And so this is extremely powerful because PNA has numerous applications as a therapeutic. It can permanently bind to DNA in your body and change host expression of proteins. And so Daneth Lee's lab is interested in actually curing diseases with PNA. But you can see how getting this into the body might be concerning if it does bind so powerfully to DNA. And so left-handed PNA has all the benefits of DNA nanotechnology, has potential to grow large structures and allow us to make interesting and powerful sensors, but fundamentally there's never going to be a risk of it binding with DNA inside the body. And so we think there's just tremendous opportunity for the next generation of nanobiosensors, but also for thinking forward to the time when we actually want to use these with the body. We already know they're not going to break down, they're not going to provoke a host immune response, and now we also know they're not going to cross-hybridize with DNA in the body. And these are like the three key problems with DNA nanotechnology, and so we're hoping that for certain applications, PNA may end up being a better building material.